Hello, our names are Mateo López and Jasmin, and this is our seminary based on the article IGF-1R axis inhibition restores dendritic cells antitumor response in ovarian cancer.
Ovarian cancer is a type of cancer that only causes symptoms when it's already spread into the abdomen and the pelvis. And there are three main types of it. It can be either an epithelial ovarian cancer, a stromal cancer, or a cancer in the germ cells that produce the eggs.
The relation between IGF-1R and ovarian cancer is the insulin-like growth factor axis is a potential therapeutic target and plays a key role in regulating growth and development of multiple cancer types. Its function is to regulate processes involved in glucose metabolism and cell proliferation. The presence of its receptor, the IGF-1R, has shown to cause a significant inhibitory effect on the proliferation of ovarian cancerous cells. Studies demonstrated a correlation between tumor-infiltrating lymphocytes and increased the EOC patient survival. Observing the IGF-1R inhibitors using immunotherapy can supply us with the understanding of the interplay between the IGF-1R axis and the immune environment in EOC.
The objective of the article was to provide evidence that the IGF-1R axis inhibition restores dendritic cell antitumor response in ovarian cancer.
An immunohistochemistry is a microscopy technique used to visualize cellular components. Immunohistochemistry allows us to reveal the existence and localization of a target protein in different cell types, biological states, and subcellular localization within complex tissues. Antibodies are used and conjugated within enzymes that catalyze reactions to form detectable compounds in order to observe specific antigens in a tissue sample. In this experiment, this test was useful to evaluate the 36 tumor samples of each case. They gave several results giving different stages. The procedure was the following. Protein expression was analyzed and correlated to the clinical data of the EOC patients, including previous hormonal therapy, tumor stage, and grading treatment protocol after diagnosis, progression-free survival, and overall survival. Western blot is a laboratory technique used to detect a specific protein in a blood or tissue sample. The method involves the use of gel electrophoresis to separate the proteins from the sample. The separated proteins are transferred from the gel to the surface of a membrane. The membrane is exposed to a specific antibody against the protein under study. Antibody binding is detected using a radioactive or chemical marker. A Western blot is sometimes used to diagnose disease.
Flow cytometry is a mechanism that is used to measure different characteristics in different types of cells. It consists of passing thousands of cells through a fluidic system, and each of them is going to be flowing down till they get struck by a laser. That striking of the laser is going to cause different light scatters depending on the characteristic of the cell, but because of its size, we're going to have a forward scatter, but because of the granularity and structural complexity of some cells, we're also going to have side scatter of that light. And to measure fluorescence, we need fluorochrome labeled antibodies that are going to target specific types of cells and then are going to cause fluorescence when they get hit by the laser, right? All these characteristics are measured by detectors that turn them into voltage, and specifically for this experiment, dendritic cells that have been differentiated were targeted with these antibodies. In the study population, tissue samples were taken from 36 patients. All of them had epithelial ovarian cancer and were operated in the same hospital. However, no further criteria apart from the time was applied to select them. A total of 10 of them had the cancer in an early stage and 26 had it in an advanced stage. But something that they all had in common was the histology of the cancer. It was a serous carcinoma, which means that the stroma was not affected.
In the results, the percentage of dendritic cells population is shown in the figure B. THP1 cells treated with IL-4, GM-CSF, TNF-alpha, and lonomycin expressed significantly higher in CD141 and CD11B levels compared to the untreated THP1 cells. Next, in the figure C, we analyze the expression of IGF-1R in dendritic cells compared to THP1. A representative Western blot analysis shows a significant decrease in phosphorylated and a total of IGF-1R levels in differentiated myeloid dendritic cells. In this figure, in the 2A, the treatment of THP1 cells resulted in decrease of IGF-1R phosphorylation levels after 24 hours. Dendritic cells differentiation was induced with or without the MVP AEW541 treatment. We then assessed the effect of IGF-1R inhibitor treatment of myeloid dendritic cells on epithelial ovarian cancer migration.
We have the results of a scratch essay. It turns out investigators took dendritic cells and co-cultured them with a line of ovarian cancer cells called the ES2, but in two different ways. Here we have a sample in which those dendritic cells did not have an inhibitor for the protein IGF-1R, but in this sample they did have an inhibitor for that protein. After co-culturing those cells, the investigators performed a scratch assay which consisted of scraping off a portion of the culture in order to see how long it took the epithelial cells to reoccupy that empty portion of the culture. They found that 24 hours later, there was a 15% reduction in the migration of the epithelial cancer cells that had been co-cultured with the dendritic cells that had the inhibitor. And 48 hours later, that reduction of the migration was of 26.5%. And those facts are very visible in this graphic and in this graphic. Here we have the results of the immunohistochemistry. We have a comparison between samples of patients that had the ovarian cancer in an early stage compared to some that had it in an advanced stage. We could easily think that there is a much bigger presence of all the antibodies in the samples with advanced stages because of the colors, but the reality is that there was only one case in which they found a significant correlation between the presence of an antibody and the progression of the cancer, which was the case of the IGF-1R. And that's because the value of the P was of 0.05, which was a significant result.
In the discussion, the author Smith said, insulin-like growth factor, I regulation of immune function, a potential therapeutic target in autoimmune disease. He agrees with the author because he is recognizing that the IGF has a key role controlling immune response. It's just that autoimmune disease are believed to be solved by helping it do so, and the ovarian cancer by blocking it from doing so. Next, the author Leo said, the stimulation of IGF-1R expression by Lewis antigens provide a powerful development mechanism of epithelial ovarian carcinoma. He agrees because he's implying that the expression of that protein makes it easier for the cancer to grow up, as the author said. For the last one, the author Joan said, function of insulin-like growth factor 1 receptor in cancer resistance to chemotherapy. He agrees because he accepts that the IGF-1R expression helps cancer survival, as the author did.
As conclusions, we have that molecular techniques are very important in medicine to provide evidence for hypotheses built off of clinical observations. Also, that the use of a control sample is very important in every experiment to identify the actual cause of the change in a variable. And finally, that molecular techniques are ones most relevant investigations cannot be carried out well without.